Okay, thank you. Good afternoon, everyone. I'm Yumu. It's my honor to introduce Professor Yixian Wang to you. Professor Yixian Wang is an associate professor in the Department of Chemistry and Biochemistry at California State University. She received her bachelor's degree in materials chemistry from Peking University in 2006 and the PhD degree in chemistry from the City University of New York in 2013, where she worked on analytic chemistry with Professor Michael Merkin. Professor Yixian Wang then worked with Professor Nongjian Tao at Arizona State University, where she focused on plasmonic-related applications. After finishing her three-year postdoctoral position in 2016, Professor Yixian Wang joined the faculty at California State University. Now, her research interest centers in developing novel analytical imaging and sensing tools for a broad range of applications, such as biomedical, nanomaterials, and environmental science. Today, Professor Yixian Wang would like to give us a presentation on single nanoparticle electrochemistry using plasmonic electrochemical microscopy. Let's focus on Professor Yixian Wang again and enjoy her talk.
Thank you very much, Yu, for your nice introduction. And I would like to thank Paula again for inviting me to do this presentation at your center. Can everybody hear me clearly and see the projected screen? Okay, great. Thank you very much. So today I will be talking about single nanoparticle electrochemistry using plasmonic electrochemical microscopy. I chose this topic because this is what we have been doing, and it might have some connection to what your center is interested in doing. The other projects we work in the lab is more towards the biomedical field and environmental science. So that's the reason I choose this one. Now, for today's talk, it will be including the three parts. First, I will talk about what is single nanoparticle electrochemistry, and then a brief introduction to plasmonic electrochemical microscopy, the technique we have been using in the lab. And then I will give you two examples. One is on the gold nanoparticles, and the other one is on Prussian blue nanoparticles. So using PEM to study the electrochemical activities of those nanoparticles at an individual particle level. First of all, why are we interested in single nanoparticle electrochemistry? We know that nanoparticles have been widely used in energy research, catalysis, and biomedical fields, because of their special chemical and physical properties, likely due to their small size, but large surface to volume ratio, and other specific chemical properties. Now, typically, nanoparticles are studied using assemble methods, meaning that we usually study bulk nanoparticles as a whole, and as a result, you will get averaged information. But on the other hand, I'm showing you an AFM image of a batch of Prussian blue nanoparticles was synthesized. So it's very obvious that with the same batch of particles, you can see different sizes, different surface structures, and we believe that the size, geometry, surface structure can all affect the actual electrochemical activities of nanoparticles. And if you use a sample method that can only give you average information, you cannot establish a correlation between the structure, specific structure of the nanoparticle and the induced electrochemical properties or catalytic properties. So this is why a new field was generated. People started to develop methods, techniques to study nanoparticle electrochemistry at a single nanoparticle level. And we call it single nanoparticle electrochemistry. And sometimes you would see terms like single entity electrochemistry, which also is related to the topic I'm talking about today. Now, before I go to the specific technique we use, PM, I would like to give you a brief survey of the existing techniques that are available for single nanoparticle electrochemistry. Starting from the traditional electrochemical techniques like cyclophotometry and amperometry, you can use a nanoelectrode to detect a single nanoparticle. So as I shown here, if you have a nanoelectrode, you can attach a nanoparticle to it. And as a result, the electrochemical signal you can observe is due to the nanoparticle instead of the electrode. Other traditional electrochemical techniques involve the scanning probe microscopy techniques, such as scanning electrochemical microscopy, scanning electrochemical cell microscopy. So for both of these techniques, it's still based on electrochemistry, based on current measurement, but they're using a nanoelectrode to scan over the surface. So as a result, you can get a spatial resolution of the sample for different nanoparticles present on the surface. The good thing about traditional electrochemical methods is you get the current signal directly. So it's straightforward. However, the limitation is that you can get one particle at a time using a nanoelectrode. So the throughput is relatively low, which is why people started to look into optical imaging techniques, which can provide you high throughput because we're just taking images, right? By taking images, you can look at different particles simultaneously, unlike the scanning techniques. So there have been publications on using different optical imaging techniques, such as fluorescence microscopy. Well, you have to use a fluorescent label, or dark field microscopy, which utilize the scattering at the nanoparticle to associate the signal with its electrochemical properties. And furthermore, the people work on the electron microscopy techniques have developed the in situ electrochemical cell, which can be used with DEM or SEM. So they can look at very high resolution nanoparticle structure alteration during an electrochemical method, electrochemical measurement. So if we have to summarize those different techniques, the traditional electrochemical techniques provide direct measurement of the current. However, they are lack of high throughput. Optical imaging techniques are can provide you a very high throughput, but the problem is more towards the diffraction limited spatial resolution, which is typically at hundreds of nanometers level. Now, TEM can provide you very high resolution, but of course the operation cost is very high and it requires specific training for using those techniques. So let's move to the technique we're using in the group. It belongs to optical imaging techniques. So the technique we used is based on surface plasmon resonance, which is a phenomenon that utilizes the collective excitation of electrons in a thin film. For example, the most widely used material would be a gold film to probe small changes occurring at the solid solution interface. So SPR itself is a spectroscopic technique that has been widely used for a long time for biomedical studies. In the case, for example, molecular interactions such as an antibody-antigen interaction. Here I'm showing you a typical setup for a traditional conventional SPR spectrometer, where you have a gold surface and we have incident light coming to the gold surface. At a specific angle, the light will be absorbed by the gold surface to generate a surface plasma. And we call this angle a resonance angle. When you have changes ongoing on top of the gold surface or near the gold surface, which is the sensor plane of SPR, the resonance angle will go through a change. We call it an angle shift. And through the detector, you will see that the resonance angle moves from 1 to 2. And this angle shift is used to monitor any molecular interaction reactions at the sensor. So this is how a conventional SPR works. Then people started to build up imaging techniques based on SPR. And by utilizing a prism or a high resolution objective, you can use p-polarized light and collect the images through a CCD camera. Now, in this case, you're no longer monitoring the angle shift because a p-polarized light is used. So all light are at the same direction at the specific angle. Now, instead, you're looking at the intensity change occurring at the same SPRM incident angle. So this is the delta I. You will get images, and at each pixel, you're looking at the intensity change, the delta I here. And within a certain range, it is believed that the intensity change is proportional to the angle shift, which can be directly correlated to the reactions going on the surface. So this is a difference between conventional SPR and SPR microscopy technique. And then SPR can be combined with electrochemical measurements. It has been used for a long time for traditional SPR setup with no imaging capability, as we call it electrochemical SPR or EC-SPR. In this case, you simply use this gold surface as a working electrode. Now the gold surface is working as a surface as an SPR sensor, but at the same time, it will be used as the working electrode, and you have the reference and counter electrode inserted. So there will be an electrochemical cell built on top of the SPR detector. And this has been used for different applications as listed here. Now, if you do a small change and move it from a spectrometer to a microscope, you can get electrochemical reactions mirrored with the images. So we can get image sequences, and within the image sequences, we can look at different area of the sample and look for individual particles and extract the information and those individual particles. So this is actually the setup we have been using for our ongoing research. Now, when I talk about the techniques, or if you read it in the literature, you might notice different names have been made there. Sometimes people call it Plasmonic Electrochemical Microscopy, and you have Plasmonic Electrochemical Impedance Microscopy, and sometimes we call it Plasmonic Electrochemical Imaging. But regardless of the different terms, we're all talking about the same concept, which is you combine electrochemical measurement with a surface plasmon resonance microscopy setup. So in my talk, I will just name it as Plasmonic Electrochemical Microscopy. It's a microscope utilize plasmonic signal to probe electrochemical reactions. Now, this technique was first reported by the late Professor Nongjian Tao, who was my postdoc advisor. Unfortunately, he passed away in 2020 at the age of 56. So in 2010, he published the first paper utilizing SPR imaging setup to probe electrochemical reactions. He used several examples. One of them is to use the imaging technique to detect the TNT residue in a fingerprint. So here you can see that the red line represents for the traditional electrochemical cyclophotometry, and the dotted line are extracted from SPR images, and they show a very good fitting, which is the first demonstration of using this surface technique. We can indirectly gain electrochemical reactions at local local positions. My talk will focus on one of the applications, which is nanoparticle studies, because we have this imaging capability. So it can be utilized to look at multiple nanoparticles at the same time. And very early report using this technique for nanoparticle studies is also from Dr. Tao. And Dr. Shem is actually currently in University of Houston. So they used the PEM technique to probe the hydrogen evolution reaction at individual platinum nanoparticles. And you can see here, those individual tails are characteristic signals that you can see from TEM images originating from scattering of a nanoparticle of the plasmonic wave. And you can think it as a boat moving in the river. So you will see those waves behind the boat. Now the difference is that the particle is not moving, but it's the plasmonic wave that is moving. But anyway, we usually will see a long tail behind a nanoparticle, and we actually use this tail to extract signals for probing nanoparticle because as I mentioned earlier, optical imaging techniques are subject to diffraction limits. So you have a spatial resolution at about 200 nanometers, but we can actually use the technique to look at particles that are much smaller. For example, I think in this paper, they claim that 8 nanometer platinum nanoparticle can be probed. So this long tail behind each particle is very important for you to be able to identify the particle being there. And at the same time, the intensity of the tail also give you information about how big the particle is. There's definitely a drawback of that is that if you have a lot of particles on the surface, the tails could be interfering with each other. And a colleague from Dr. Tao's group actually developed mathematical methods to deconvolute the overlaying of different tails. Now, this is a general idea of how the tails are generating. And then if we want to be more mathematic, like how do we describe the signal from individual nanoparticles? Here's an example, and you can see that the intensity of individual particle is actually a combination of the partially reflected light and the scattered and the plasmonic wave scattered by the nanoparticle, and it's affected by the scattering property of the nanoparticle, which is associated with the optical properties. For example, refractive index. So when you have an electrochemical reaction going on the nanoparticle, causing the refractive index change to increase or decrease, you will get an intensity change. And also, because we assume most of the time the optical change, the property change is proportional to the electrochemical reaction per time, by simply using this relationship, we sometimes assume that by simply taking the derivative of the intensity change, derivative of the intensity at the local particle, it's proportional to the current density change at that nanoparticle. So without doing a very complicated conversion, the SPR image can be converted to a plasmonic current image, which can be correlated to electrochemical reactions. Okay, so this is a brief introduction of how this technique works. And then I will start talking about the two examples using this technique for study single nanoparticle electrochemistry. The first part is what I did as a postdoc in Dr. Tao's lab. And we use this technique to study the surface electrochemistry at gold nanowires. The system is very simple. We're simply looking at the surface of the gold particle, conversion from gold to gold oxide, and then conversion back from gold oxide to gold. The original motivation of this work is we would like to see, because we know the surface reaction is sensitive to the surface facet of the particle, so we would like to see whether we could get structure information out of serving different combinations of nanoparticles. Now here I'm showing you two videos. The first one is the oxidation process of the gold nanowire. So you can see that each tail represents a gold nanowire. The wire is sitting at the top of the tail. And you can see that when the oxidation going on, you see the intensity increase. And then this is during the reduction. Okay, it's an opposite signal compared to the oxidation. And we can compare it, we can compare the SPR signal to the potential stat CV, which is collected directly from the potential stat that induce the voltage and record current. So here shows the black one is the conventional CV, and the red one is the plasmonic CV. So I believe that the red one is the plasmonic CV with nanoparticles, and the blue one is without particles. So you can see that without particles, we don't have any current, and with particles, we see the oxidation and reduction peak, and the position overlays very well with the potential stat CV, demonstrating that we can actually visualize the surface reaction at individual nanowires. And then we can extract information from different nanowires. So for example, we take 10 of them here, and from the optical image, you can estimate the size of the particles. And then from the PEM, you extract the current signal. And you can see that different particles kind of give you different intensity and slightly drift in the potentials. We did a little comparison of the intensity among different particles correlating with the size. There's not a clear linear correlation between the size and the signal. So which means that it's not necessarily that when you have a larger particle, you will have a stronger intensity, current intensity, which is probably due to that the particle surface is covered with some capping layer, which cannot be fully removed during the experiment. So some large particles cannot give you enough electrochemical signals. Now, we also try some even longer nanowires. So in this case, it's a 10 micrometer nanowire. And we can observe that with this technique, we can observe that within a single nanowire at different position, different location, like at the two ends of the particles, you actually see different electrochemical activities. So this could be due to that the surface facets is different at different position of the nanowire, or it could be that the capping layer is putting an effect there. But in either case, to be able to fully establish the correlation between surface structure and its electrochemical activities, we still need to incorporate other techniques to provide us more structured information in this case, which is something we're still trying to figure out at this moment. And now, if you still remember at the beginning, earlier I mentioned that our original motivation is that we would like to see whether by serving different particles, we can kind of see a correlation between the surface facets and the electrochemical activities. Now, this starts because we have surveyed the literature that when you have different surface facets, the surface electrochemistry can actually provide you a lot of information. So for example, this paper talks about using the single crystallized electrode with 111, 110, or 100 different type of surface. You can actually get different minor peaks in a CV. And those different minor peaks can be used to identify different type of surface. So what we have been trying to do for a long time is whether we can get a nanoparticle electrochemistry CV, and from that specific CV, can we look for those identification fingerprints that can correlate to the surface facets, so we don't have to do a TEM or other technique for identifying the surface structure. And we know that different particles are composed of different facets. So in the next work, we tried different combinations of, we tried nanospheres, we tried nanocubes and nanorods, which can have different ratio of 100, 110, or 111 surface facets. I'm not showing you all the results here because I don't think it's fully conclusive. It's still a topic that needs further investigation, but I'm showing you one example here we did with gold nanorods. So with gold nanorods, we did some TEM and noticed that with the same batch of nanorods, we can see that some rods are more round and some has very clear facets. And when we obtain the CVs from individual nanorods, we actually observe that different nanorods have different minor peaks for the oxidation part and for the reduction part. So here we show one example. The different color actually represent for different cycles. So we run 4 cycles, and this is another interesting we observed. Even for the same particle going through the electrochemical reactions, we see that the peak is not staying at the same location, which might suggest there could be a surface structural dynamics going on there. And here we shows that we surveyed multiple nanoparticles and comparing the peak positions from cycle 1 all through cycle 4, and you can see that there is a general drifting from about 0.26 volt to like the disappearance of this high minor peak around the same potential. And same thing here, we also see some systematic transformation of the minor peak positions during the cycles. Now, I cannot say that this is enough to indicate that we demonstrate the effect from the crystal facets and surface structural dynamics, but it's definitely a beginning that we're able to probe different nanoparticles within a same system, observe the difference. And as I mentioned earlier, it would definitely be helpful to combine with other techniques to further correlate the electrochemical activities with its structure. Now, the next project that my students have been working on is another simple system, which is Prussian blue nanoparticles. So Prussian blue nanoparticles was originally developed as a dye molecule, and it's composed with iron 3, iron 2 atoms with cyanide groups. It is a very widely used material for a lot of field, and more specifically in electrochemical sensing, it's a nice catalyst towards hydrogen peroxide reduction, which is why we started looking at this particle and wonder whether we can get some useful information utilizing the PEM technique. Now, specifically for the hydrogen peroxide reduction, the insoluble type of Prussian blue, the Prussian blue can have insoluble or soluble forms. And here I'm showing you the insoluble forms as, for example, Prussian blue nanoparticles. And if you induce a reduction on the Prussian blue nanoparticles, it can be converted to Prussian white. And this Prussian white has a catalytic effect towards the reduction of hydrogen peroxide, which will in turn convert the Prussian white back to Prussian blue. The good thing about this system is it has a very dramatic color change associated with Prussian blue to Prussian white. So PEM has a very high sensitivity to this reaction. The first part we try to do is we survey in the literature and look for different methods of synthesizing Prussian blue nanoparticles, because that according to literature, you can use different methods to control the geometry of the Prussian blue nanoparticles that are synthesized. Now, in this case, we used 2 different methods. One is simply combining iron 3 thought with the iron 2 thought. So it's a direct synthesis. The other one utilized PVP as a controlling agent. So it's a slower process. It can gradually decompose the potassium ferrocyanide existing in the solution, and then the iron 3 will combine with the iron 2 with the presence of PVP, so that they call it surface control methods. And with this method, you're expecting to synthesize cubic shape like Prussian blue nanoparticles. Well, the first one is more random shape, which will be generated. And then we use AFM to demonstrate the product. We synthesize with either method. And it's further confirmed that from the type 1, it's more randomly spiral shape, while the type 2 has more cubic or more sharp edge structures. So then we apply these two different types of particles and see whether we can get different types of signals because their structure are different. This is the first example that we used PEM for the type 1 Prussian blue nanoparticles. So as I said, so this one has no QAP, so the product is more random. And specifically in this example, we used some gold chips that were etched with some patterns. So it will make it easier to later correlate the location of the particles. And here's a video of the PEM images, which you can see that we have the particle spots, which give us the Prussian blue to Prussian white electrochemistry. And here is the potential stats CV we obtained from the electrochemical setup. And here are two examples of Prussian blue nanoparticles. There's one here, and its surrounding background area. So you can see that we got very nice symmetric peak, which correspond to the thin layer electrochemistry of Prussian blue coated on the electrode surface. And if we overlay the potentials, the peak potentials of those Prussian blue nanoparticles with the potential stats CV, we can see that the peak position correlate very well with the potential stats CV, demonstrating that we are observing the electrochemistry of Prussian blue nanoparticles. And we got a wide range of intensity distribution, which can give us information of the size or the activity of different particles. So this is type 1. It's very straightforward. We got what we expected. Now with type 2, we expected to see something special because those particles are more cubic shape, but unfortunately what we see is a very weak signal. And here's the example. So here is the potential stats CV, and you can see that it deviated from the symmetrical type of shape, which we can expect it from a thin layer from Prussian blue already coated on the electrode surface. And then when we extract signal from individual particles, we got very distorted CVs, and the signal is much weaker than type 1 Prussian blue nanoparticles. We also tried varying the amount of PVP present in the initial synthesized method, and these two are with 3 grams PVP, and this is with 10 gram PVP. And you can see that with 10 gram, with more PVP present, you have even more distorted CVs. So we think that it's probably due to the PVP residues on the Prussian blue nanoparticle surface that prevent the potassium ion getting in and out of the lattice to realize the reaction. And at the same time, it might block the contact between the Prussian blue nanoparticle to the gold surface for the electrochemical reaction. And so what my student did next is they tried to remove the PVP by ethanol, by acetone with different methods and see whether there's any improvement in the signal. I haven't included the data. It's more recent data. I haven't included it here. We do see some improvement, but it's still not as ideal as what we want. So we're still looking for better methods to solve the problem. And then finally, we also did some hydrogen peroxide reduction with those Prussian blue nanoparticles and trying to see whether we have those hydrogen peroxide sensing capability at individual nanoparticle level. Here I'm showing you the videos of the Prussian blue reactions at different concentration of hydrogen peroxide. It might not be very straightforward with the videos, but then if we overlay, if we put the images together at different concentrations, you might notice that there is a change in intensity when the concentration of hydrogen peroxide is different. And here we have the comparison between the potential stat CV and the plasmonic CV. And you can see that the peak is still there due to the Prussian blue to Prussian white conversion, and the peak current decrease due to the hydrogen peroxide presence, because some Prussian white were immediately pushing back to Prussian blue. And if we use the intensity here at the reduction peak, we can get linear plot, which is pretty good for hydrogen peroxide sensing at an individual nanoparticle level. So that's the example I would like to show here. And the project we're currently working on is to develop some other modes of PEM analysis that can realize some other capabilities. So one of the example is, because I mentioned that Prussian blue system has very dramatic color change, and we would like to use that to build out a system which can provide more sensitive detection toward hydrogen peroxide or other oxygen reactive species that can be released from cells or other systems as an imaging system. Now, from the examples I've shown, that we're still having challenges using these techniques, for example, for more complicated systems like batteries, food or environmental samples, which you will have large background interference, and how do you get informative information from those type of system. And the spatial and temporal resolution could be further improved. As the imaging technique, we always talk about the diffraction limitation, and that's something we have to overcome. And then finally, with the PEM technique or SPRM technique itself, it's not enough to provide the information we wanted. For example, for correlating the structure with electrochemical activities due to the limitation in a lot of aspects, such as spatial resolution. So being able to combine SPRM with other techniques is another thing we have been working on. At least we would like to be able to correlate it with AFM measurement. That's something we're currently working on. Okay, so that concludes my talk, and I would like to thank my students. This is kind of the old crew before the pandemic, and this is after the pandemic. And the work where I'm showing here is specifically contributed by Fatima, Adelie, Kingsley, and Mariam. And I would like to thank to my collaborators, Professor Tao, Professor Wang, and Professor Shen. And also I would like to thank the support from Cal State LA and NSF. So that concludes my talk, and thank you very much to everyone for being here today. And any questions?
Thank you so much for the great talk. Are there anyone holding any questions or comments? Please feel free to raise your hands. Oh, Dove, go ahead.
I should wait a little bit for students to ask first, and then I can ask later.
Danwei, go ahead. That way, that might warm up the students.
All right. So Yixian, it's really cool technique, very nice work. I'm curious for this imaging, SPRM imaging. Have you done the experiments for the nanoparticle to hold the potential constant and then observe the evolution of the plasmonic spectrum?
Yeah, that's a very good question. So that's actually something we are doing. It's just because of the pandemic, the process has been delayed. So for example, for the Prussian blue nanoparticle I would like my student to hold the potential and see how it responds to hydrogen peroxide. Unfortunately, we don't have a presentable data right now. And also for the nanoparticle systems, we're also working on other problems, and definitely amperometric detection would be something useful.
Yeah, that's awesome. Is it true that you primarily work with undergraduate students, or do you also work?
Yeah, so the picture I'm showing here, I have only one or two master students, and all the others are undergraduate students. Yeah.
All female team. Kudos to that.
Yeah.
I have a question. Not sure there is a good answer for it, but do any of these techniques apply or like could be applied to molecules like homogeneous? Is there a way, or like unless you support the catalyst, I assume it's difficult to do any of this cool techniques?
So I'm trying to understand your questions. Are you asking about molecular catalysts?
Yeah.
I believe that it would be hard to look at individual molecular catalysts because they're too small. Yeah, it's the signal. You might be able to see overall signal change, but it's hard to contribute the signal to individual catalyst.
I thought so, but I thought I might ask anyway.
To add on to that, actually, what about like proteins?
Proteins is actually something people have worked on. SPRM technique can be used to look at protein structure transformation. My group didn't work on this area, but I think late Dr. Tao's group, they have some work on single proteins, and they single protein, they tried it. It's probably a little bit difficult, but at least they did single DNA analysis, virus analysis, and bacteria analysis, which is much bigger, of course.
Any questions or comments? Oh, Jeff, go ahead.
Thank you for the great talk. It's a really interesting technique. I want to echo my colleagues here. I think this could be a very powerful method for identifying, aspect ratios that are awesome for catalysis. And so I know that you want to target like really well-defined particles at the onset, but is the idea then to apply this to like a really dirty synthesis of materials that may have a, aggregate mixture of different things so that you can then pick out, oh, these guys are really, really active species. And then using your AFM, perhaps you can then get information about, structure or composition. Is that the idea, or am I missing something?
Yeah, that's definitely correct. So one thing we would like to achieve is we do have an AFM. The problem is that the current SPRM image area is much bigger than the AFM range you can image.
Yeah.
We're looking at hundreds micrometer level, and for AFM, the maximum we can do is probably 50 micrometer. So it's hard to do a complete one-to-one racial mapping. But if you have a sample surface and you have some specific area of interest, it's definitely this technique can provide you heterogeneity information. So I can see this area is more active, that area is less, and we can use the AFM to look at the specific areas. And another thing, when I talked to Chong, who is not here today, he mentioned that you guys are interested in doing polymer synthesize, and he asked me whether we can look at the polymer patterns. And I asked him the size. He said is around 100 micrometers, which is definitely okay with our system. And I mentioned to him that our department also has an IR microscope. So once you have the polymer pattern, we monitor the reactions, and then we can move it to AFM and to the IR microscope, which can provide you chemical information. The AFM will provide you mechanical information.
Thank you.
Sure. Thanks for the question.
Any questions? All right, if there is no questions, thank Professor Yixian Wang again, and thank everyone for participating in the webinar. So CSA members, please stay here for our next session. Others, please feel free to leave.